postdoc in our group, and I'll tell you a little bit about Carol. So Carol has come to work in our group on a Madame Curie fellowship called Nitrogen Regeneration Under Changing Oxygen Conditions. But this is only after she received a PhD at a joint program between Max Planck and the University of Kiel, where she spent another 3 years or so as a postdoc. And Carol, she, it's funny, she has a little mind map thing about what she's interested in. And I just was looking at this because it's kind of cool, you know, you can sort of take something that you've written and sort of do an analysis. And the main thing that she's interested in isn't on here, which I think is really weird, because if I were to summarize what Carol's interested in, it's nitrogen. Nitrogen in the environment, and especially nitrogen in the oceans. And that's actually critical because nitrogen is a key component of life. And there are processes in the oceans which take nitrogen out of the waters. And what Carol has done is she has come up with new methods to understand those processes which put it back, which allows life to persist and survive as we like it, as we know it. So I think that leads us up to the title of the talk, is Does the Ocean Lose Its Breath, which might have something to do with that. I guess we'll
You never
see.
know if nitrogen is coming up or not, right?
You never know. But anyway.
Yes, thanks for the introduction. I don't know why nitrogen is not on there. It's just how it happened. So yeah, thanks for your introduction and thanks for inviting me to that lecture series where I can present on my topic more or less, Does the Ocean Lose Its Breath? And I'd like to frame that talk in a certain way because I want to dedicate it to my former colleague, Victoria Burtix, who I was working with at Geomar for 2 years or so. We shared an office, and Vicky was actually not only my colleague but also my friend, and she was one of the brightest young scientists that you could imagine, and was always a great partner for all discussions. I guess some of you know her. And she was like posthum awarded a couple of honors and prizes from the AGU, from the ASLO and other things. And I think it was a great loss for all of us at the Geomar, her friends, and for biogeochemistry as a research field that she passed away that early from breast cancer. So anyway, we were working quite a bit together, and we were doing this mainly by going on joint cruises. We spent quite a lot of time on this research vessel. It's a German research vessel, Meteor. And we traveled the ocean in order to find out about biogeochemistry, about climate change, and how the ocean responds to it. And we were in a lot of ocean areas, starting from basically the North Pacific to the South Pacific. I was with Vicky in the Atlantic, the Baltic Sea, Spitsbergen was a bit cold. And then we were also in that nice tropical area that was very enjoyable because we did a couple of dives afterwards. And Vicky saw some whales, which she always liked. And I just calculated yesterday how much time of my life I actually spent on research vessels, and it's really more than 1 year of my life that I was on a ship in order to collect my data, to do my research, and I just did that in order to understand the impact of climate change on this whole system, on the ocean, and how it would respond if climate change gets stronger and stronger, also regarding the nitrogen cycle, not to disappoint you. Yeah. So whenever we went to cruises, Vicky and I and also others, we looked at the data, and our data is a combination of different kinds of. We, for example, just profile through the ocean in order to collect data on oxygen, on nitrogen species, nitrate, nitrite, to see what the nutrient status is, and also like iron or other trace metals in order to understand what controls the productivity and what controls the ocean as a system. And we also measured a couple of rates. We looked at the organisms, the bacteria, the microbes, using metagenomics to understand what metabolic potential is in the ocean. And whenever we were discussing about how the Earth and the ocean respond to changes in all of this, Vicky was strongly arguing that the Earth is more than just a planet, but it is a living system, and we are all parts of it. And so are all organisms on Earth. And this is the famous Gaia hypothesis, which was put up by James Lovelock and also by the Margulis, of course. And we were kind of in love with this thought. And whenever we were discussing whether this could be, whether actually the Earth as a system is able to respond to climate change and to basically regulate itself in a way, Vicky brought up this figure, which I personally don't think is the very best example to explain that. But as she loved it so much, I will show it to you. And it basically shows the CO2 development over a geological time scale of 500,000 years or so. And what you can see is how CO2 basically changed in concentration on Earth. It went up, it went down, up and down again, and up and down. And Vicky was always saying, see, whatever happens, whatever CO2 increases on Earth, the Earth as a system is able to kind of counteract and to get it down again. So it is, for her, this was basically a proof that the Earth is able to interact or to counteract and mitigate climate change on its own. And then I was always coming and saying, like, but look at that. That's not going to work out forever, right? So, and obviously it is not, because CO2 levels are increasingly going up. And this is even from 2007, where it was below 400 ppm. But now we already crossed this mark. I guess it was only a couple of months ago that we went over this 400 ppm. And at that point, we cannot be sure whether the Earth as a system or the ocean as a system is able to, at any point, counteract any more to that. So this extreme increase is obviously human-made, right? It comes through fossil fuel burning, and this leads to CO2 output to the atmosphere, which then increases global temperatures. And this has a lot of severe consequences, and one of which is obviously the melting of the polar caps. And that is a consequence which, at that point, I do not believe can be mitigated anymore by a system like the Earth we see. That needs a bit more. But then on the other hand, and this is my major interest, also the ocean, due to global warming, loses oxygen. And this is for several reasons. One of the reasons is just like the chemical solubility of oxygen, because if it gets warmer, oxygen dissolves less good in water. But also you have an increased stratification in the ocean, which means you have a top layer that warms up, and oxygen cannot penetrate to the deeper waters anymore, so there is less of an exchange. And the third point to it, why oxygen gets lost from the ocean, is mainly because the ocean circulation is also changing. And oxygen is not that well transported to the ocean anymore. So, at that point, like Vicky and I were always kind of disagreeing. She was still of the opinion that if we just look at it for a long enough time scale, we would find that it will mitigate the Earth on its own climate change. Well, I said that we can discuss it forever, it doesn't make sense, we just have a look at it. And so we had a look at oxygen distribution in the ocean. And this is what we find in our current oceans. That's oxygen distribution at a depth of 200 meters in the water column. So if you dive 200 meters deep, you would find something like this. And you see in blue, low oxygen concentrations, and reddish colors show high oxygen concentrations. What you can basically see is that there are a couple of ocean areas which are pretty depleted in oxygen. The weak oxygen minimum zone is here in the Atlantic. Stronger ones is here in the Indian Ocean, the Arabian Sea, and the Bay of Bengal. And the most impressive, the large and persistent oxygen minimum zone in the Pacific. Yeah, that's obvious. The biggest thing, obviously the biggest thing that we have in our ocean. And this is also the area where Vicky and I and other colleagues did most of our studies at. In the same paper that this figure is taken from, they calculated from a 50 years time series study whether the oceans could lose oxygen in the future or not, and if so, where. And what they found, and this is here indicated in blue, is that there are huge areas where oxygen is further lost from the ocean. And these are the areas where we already have not so much. So this would basically mean that oxygen minimum zones, oxygen depleted areas in the ocean would expand and get also more intense in their anoxia. So this means that basically oxygen is largely lost from the system in exactly that areas that do not have oxygen at that time already. So this is still based on only a certain time frame. This whole study is based on data collections over the last 50 years, because there is not much more, because people did not collect data before. And this means that the time scale of this whole calculation and this whole prediction may be a bit short. So we basically were still wondering whether this is the truth or whether this is only a temporal effect, and we wanted to know how the ocean overall would respond as a system if oxygen gets shorter, and whether basically there are organisms or there are feedback mechanisms that could counteract this. And we thought about, okay, thinking of the ocean as a system, like we did about the Earth as a system, we need to think about what parameters are important. And this obviously starts where life starts in the ocean, with primary production. The primary production takes place via phytoplankton, little plant-like cells that produce from sunlight biomass. And as soon as you have production of biomass, you also do have cells that die. And if they die, they sink down, and this is called export production. So you have basically a transfer of living matter to dead matter, and this dead matter is transported down and is respired, and you can see how this works. It's aggregating in particles, and there in yellow you have small bacteria sitting on top of it, eating it up again and releasing the organic material back to the water. But this consumes oxygen. And this oxygen consumption has certain consequences. And one of it is nitrogen loss from the system. Here you go. Right? Because nitrogen loss is a very oxygen sensitive process and takes only place if oxygen is already pretty depleted. Another consequence of oxygen depletion is that you have a release of iron and phosphorus, which are both elemental substances for life from the sediments. And the lower the oxygen gets, the more of those substances are released to the water, and that fertilizes the above waters. And this allows a certain process to take place. And now, this is nitrogen fixation, obviously. And nitrogen fixation, as part of the nitrogen cycle, is so crucial for life in the ocean as it could just be, because it allows life to grow, because all life consists largely of nitrogen and needs it. And the problem with nitrogen is it is not just there, it is in the atmosphere. And it is incredibly difficult for organisms to get this nitrogen out of the atmosphere into their body and then release it to a system. But without that, there is no life in the ocean. So, and there is where nitrogen is really important. And this basically means that you end up with having a feedback of primary production, export production, oxygen consumption, that then releases nutrients and allows nitrogen fixation to take place, because it needs phosphorus and iron largely, and then fuel the chain again. And this could run on forever, as it seems from that picture. But the problem is, the more export production you have, the more oxygen depletion you have. And this can go in an entirely wrong direction, because if you have too much oxygen loss, then you end up having a really intense anoxia. And this may be a case where you have phosphorus and iron not coming out of the sediments again, because this situation would bind them to the sediments. That's just something I will explain later on a bit more in detail. And the problem is, if you don't have any more phosphorus and iron, then you don't have nitrogen fixation, and the whole chain breaks down. On the other hand, this could mean, in terms of oxygen depletion from the ocean, that if this chain breaks down at a very intense anoxia, that the system is able to recover them to a less intense anoxia. And that would mean that the ocean could basically self-regulate its oxygen content. If you look at it as a sketch of an oxygen minimum zone, it would probably look like that you have your iron released from the sediments, it's transported to the nitrogen fixation zone, production chain goes on, and you have it running until the system gets really anoxic, and that would look like this. And in that case, you would have iron precipitation to the sediments and no more nitrogen fixation production, and so on, and the system could recover to moderate anoxia. So basically, what I was proposing then, together with Don and others, that there exists a feedback regulation based on iron, on the oxygen minimum zone intensity, and on nitrogen fixation, which could regulate the oxygen status of the whole ocean. And that would be a cool thing, because if this would happen, this would indeed mean that oxygen minimum zones do not expand forever, that the oxygen is not lost forever from the ocean, but that there is a certain limit to it. And that oxygen minimum zones would rather oscillate instead of just spread throughout the oceans, and the ocean would then die. And I think that's quite a point. So we have to have a look at that. We had to have a look at that because this was only a hypothesis, and we did some studies of Peru where we have this intense anoxic zone. And I'm going to show you just like one example on nitrogen fixation where we can actually see that this feedback mechanism could work in a nice way. And this is the area we went to, so it's right off Lima. Lima is supposed to be here, and we had a cruise going out a couple of times at a certain section that we always sample. And we had a second cruise around here. But I'm going to present the data set from that section. So what you have in that area is the oxygen distribution. This is the shelf, so the sediments. And here you have the coast, here's the open ocean. And this is where you dive down over the first 1,200 meters of the water column. What you can basically see is that you have only oxygen in this very, very thin layer on top of the ocean. In red here, it's hardly visible actually. And afterwards, a bit deeper down, you have this huge blob of an oxygen minimum zone with really zero oxygen conditions, with real anoxia. And in this yellow box, we have a sulfidic patch where actually the anoxia is already toxic and where we predicted that our system should actually break down already. And that's what we tried to find or to look at. So we wanted to basically see what happens in the oxygen minimum zone and in its sulfidic parts. And we first had a look at the iron distribution because we postulated that iron release from the sediment is one major point to that. So when looking at the iron, we saw that one. We saw actually pretty large amounts of iron coming out of the sediments in the anoxic zone, and that is fine, that is what we expected actually. So this should actually fertilize nitrogen fixation, also primary production. But what we also found was pretty high iron concentrations in the sulfidic patch, which was actually kind of not what we expected to see, because we predicted that here the iron should precipitate and go back into the sediment, but we had just a closer look. So we basically wanted to see, as a next step, how nitrogen fixation as a process in terms of rates behaves in that area. And so we did, and I measured a couple of rates. It's not so difficult, it's not so easy to get those rates actually. So I measured them over the top 300 meters of the water column. It's a pretty complicated method, so the resolution is not as high as for oxygen. But what we found was exactly what we kind of expected. We found high nitrogen fixation where iron was high and oxygen was low. But again here, we found the highest nitrogen fixation where we had the sulfidic anoxia, the real intense anoxia. And this was actually kind of counterintuitive of our hypothesis. So we could not really understand how this could be, because this intense anoxia should basically not allow nitrogen fixation to occur. So this would indeed speak against our self-regulation idea, and that would be a bit sad, right? So anyway, we thought maybe we just don't have the right concentrations, maybe this anoxia is not intense enough, maybe we did not have hit this turning point that we need to hit to see a self-regulation coming up. And so we had a closer look at the organisms that are there, because what else if not alive that sustains this whole process should give us information about that. And this is the community of nitrogen fixers in that area. And I don't expect you to read any of this, but you can see probably that there is quite a high diversity. So this is a phylogenetic tree, and you can see that every line here in this tree marks one certain cluster of organisms, one phylogenetic group. And you can see there are quite many of those. And actually most of them that are in reddish and in pink were not expected to be there, so it was a very special community. So what I wanted to do with that was basically to know which are the nitrogen fixes that you find at different parts of the oxygen minimum zone. And I selected for a couple of them, which I don't name here, but that's not important, in order to do some culture experiments. So I selected for organisms that were present directly connected to the sulfidic patch and to the anoxic zone. And I tried to do some culture experiments with those. And for this I got some isolated strains from a collaboration partner from Helsingør, Lasse Riemann, who has to be really acknowledged for that because he has an incredibly huge strain collection no one else has in the world, and it's pretty cool that he's so close by. And he gave me his strains, and what I did with them was trying to let them fix nitrogen under different low oxygen conditions and under different sulfide conditions. And the result of this is actually depicted in this figure. And what you can see here is the nitrogen fixation rates in those four cultures combined. You see the oxygen here between 0 and 15 micromolar. This is about 5% of the atmospheric value. And hydrogen sulfide at concentrations between 0 and 15. In the oxygen minimum zone of Peru, we have something like 2 most of the time. So pretty low. And what is actually quite nice when looking at that figure is that there is a clear response to the redox conditions. So you find maximum nitrogen fixation in a redox condition around 0 oxygen or slightly sulfidic. So this fits quite nicely to what we saw in the environment, actually, where we find nitrogen fixation when it's slightly sulfidic. So this seems to be still a concentration where it works out. But the other thing that is actually much cooler than that is that if the sulfidic conditions increase, if the anoxia gets so intense, then the system breaks down and nitrogen fixation does not work anymore. And that fits incredibly well to what we proposed, because it basically means that at a certain point, as soon as it's really anoxic and reduced enough, the whole productivity chain would break down. And in terms of the oxygen minimum zone, it basically means that the oxygen minimum zone intensity interacts with nitrogen fixation and primary production in a nice way, so that it kind of counteracts. The more intense the oxygen minimum zone gets, the less intense the productivity is. And this would lead to a scenario like this, where the whole system crashes at a point and is then able to regenerate because no more respiration takes place. And this speaks very strongly for the self-regulating idea that is postulated by the Gaia hypothesis, and that also Vicky was such a big fan of. The question then was like, to add something on top of that. This was only a result from the oxygen minimum zone of Peru, and we were wondering whether we find a similar metabolic potential in other areas of the ocean, because you never know, right? This was only one example. Maybe this is not valid for any other part of the ocean. Could be. So what I did was then sequencing other parts, other oxygen minimum zones in the ocean. I did a bigger sequencing approach in the Pacific area. I also sequenced different parts of the Atlantic oxygen minimum zones of Mauritania and of Namibia. I didn't show, I also sequenced the Baltic Sea and some samples from the Arabian Sea, and I will add some more sampling areas in the future to that study. But what I basically found there was this. That's a graph that is not so easy to handle, but it shows the distribution of microbes in those oxygen minimum zones. And in the very beginning, I thought they would probably cluster according to the ocean area where they are from. So in a sense that you find a cluster from the Atlantic versus a cluster from the Pacific versus a cluster from the Baltic Sea, but they did not. The major criterion they were really clustering with was the position within the oxygen minimum zone, in the sense of all organisms from the surface, no matter from which ocean area, cluster together. All organisms from sulfidic events come together. All organisms from oxygen minimum zone core waters, and from the oxyclines, which are the upper and the lower border of the oxygen minimum zones, come together. And that was actually quite impressive, because what you can learn from graphs like that is, if you have similar communities throughout all oxygen minimum zones that behave similarly in different different redox conditions, this means that in principle the same processes would be possible throughout the oceans because the potential is there. So what you can learn from this is basically that no matter which oxygen minimum zone we are talking about, the metabolic potential for self-regulation would in principle be there. And that is what you can learn from metagenomic data sets like this, although this does not include all oxygen minimum zones, and although the biogeochemical parameters are quite different there. But it strongly speaks for the fact that as soon as biogeochemical factors are similar, there could be a similar response to it. And this was, for me, quite impressive. So, coming back to Earth history, what does that mean? In principle, it could mean that if this self-regulation mechanism are functioning at a point on capacity that oxygen minimum zones would rather laterally expand and then retract again instead of just like increasing throughout the oceans, which is anyway true. Not credible to me. So we could probably rather speak about an oscillation than of an ongoing intensification and expansion, and that's quite a different story for our Earth future, right? However, we don't know like on which time scales those mechanisms really act. That's not what we can know. That's not what we can learn from our data these days, right? Because we only have a snapshot now. And although people are driving models showing that oxygen decreases, it's also only a limited amount of data from the last couple of years that we can actually do. So we can, however, look back into the past, and this is the Cretaceous, where we already had these anoxic events in the ocean, and here they are depicted in this pinkish color, and you can see it's intense, it's everywhere. And it's also due to a massive CO2 release, this time not by humans, but by volcanic activity and other things. And this triggered anoxia in the ocean during that times. The thing is, to get a feeling for time scales, one can look at the model that was calculating that, and this was a couple of 10 to the 6 years, so this whole anoxic event was quite a long thing. It was not only like a couple of hundreds of years. And so we can more or less not say whether the system just takes 100,000 or even 1,000,000 of years to recover from something like that, or whether it would rather just collapse and give evolution a chance to basically form new life from a situation where no life was possible anymore. And this is what we don't know. So to come back to this introductory slide about whether the Earth is more than just a planet but self-regulating in a way, we can basically, we have evidence that it's self-regulating. So we find in our data that there is quite some evidence for that. But as we are also part, as humans, of this living system, we need to be aware that our activities are kind of not good for that system. And a key question is basically how much can this whole Earth, the ocean, take? Where does this all not work anymore, and what can organisms, what can the Earth balance at all, right? So that's the point where we are, and we really need to think about whether our lifestyle is in a way makes us actually a part of the Earth, or rather a disease or a virus. And that is where we are. And in the end, I can only thank a couple of people, which are my major collaborators, Julio Sepulveda, Florian Scholz, and Don, obviously. Silvio Pantoja gave me access to a huge amount of samples. He's from Chile. Hermann Bange is my mentor since the last couple of years. And I thank the captains and crews, and chief scientists of the different cruises that we took our data from. And we don't know about how the Earth responds overall, and we know all that coming back to wiki life is not always fair. But it may indeed be like, and this is on a personal note, that it sustains itself in a way that where you lose something, something you can do. And while losing wiki, there was also place for new people, And I'm particularly grateful that I met a couple of interesting characters here that basically counteract this loss quite well, and it's particularly Una, Dan, and Richard, and it's a group that I want to thank because you're very inspirational to me. And that I can close, and thank you for your attention, and I'll be happy to take your questions. And if you don't find this boring, you can come to my session at the Goldschmidt conference, which will be in Paris in summer. And that's it.
Okay, let's take some questions.
Thanks for a great talk. What are the main determinants of where these oxygen minimum zones arise in the ocean?
Yeah, it's a combination of different aspects, of course. Like if you think about global warming, this of course only orders process that's already there. So it's ocean circulation for a big part. So the deep water is formed in the Atlantic, goes down, and there life interacts with it, respires oxygen away, and wherever it comes up again to the surface, it's depleted in oxygen. So this is a basic preset. But then also it's stratification, which means that the ocean has different layers. And the question is how stable those layers are. If they are very stable, not much oxygen can come through the top layer and get into the deeper waters. Somewhere where it's rather mixed, obviously more oxygen comes down. There's a couple of those factors, like physical interactions, but also like the biology depends largely on the communities that we have there, how much they respire and what processes happen. That's it.
And so can you, if for instance some currents in the oceans change, can you predict where new oxygen minimum zones would arise?
That's what people can do actually quite well. There are nice physical interaction models which are people running these days, and it was actually just a study published in Nature that showed principle a similar picture than what we had before, but based on ocean currents mostly. And so that's what works out quite well, but that's physics based, and that's not all of the story obviously. And that's still the easiest part to predict, right? Because physics cannot change that much.
Do you have some more questions?
For the oxygen minimum zones, I was just the map seemed extremely detailed. How is the method of collecting these? Is it possible to do how do you aggregate these?
So for oxygen, there is actually quite a good data set. It's one of the parameters that's always been measured since ocean expeditions were going on. But also now they have a high resolution service using remote controlled equipment, just diving up and down through the oceans, collecting data, sending it via satellites back to us, which is actually very amazing, very cool.
Yeah.
But also if you have maps like this, that's interpreted. It's never all of it. You see also for example, off Namibia there is an oxygen minimum zone. It's always there in those maps, but it's not always there in reality. It's seasonal. So that's obviously a result of the interpolation.
Some more questions? I have one or two. One is how big does the oxygen minimum zone need to get before you start into the negative part of the feedback system?
Yes, it is well, I guess there's quite a thing about the topography. Because if you think about a very productive oxygen minimum zone of Mauritania, but a very steep coastline where it's dropping down to deep sea conditions and then you think about Peru, which is not that productive as Mauritania is, but has these shallow coastlines, so it depends largely on the topography, that's what I think. So depending on where you are, this can go quite fast or not. And that's what you actually see if you compare the different intense oxygen minimum zones. But I think the lateral up and down towards expansion along the continental shelf is much more important than going out into the deep sea.
My guess is there's quite a room for expansion still, unfortunately.
Yeah, that's why we need why you need now to look into the sediment records at the borders of the oxygen minimum zone to determine whether this was the case before or not, and that's what people actually missed until now. So.
One other question. You described it as an oscillating system. But I don't see that,
Actually, it is.
huh?
Yeah.
But does it necessarily have to be? Because couldn't you actually have a self-regulating system where, for example, due to climate change, you have more stagnation, whatever, that
You can have,
actually would
yeah.
have a larger but stable extent of oxygen minimum zones?
Yeah, this would also be something you could think that exists, right?
Yeah,
But that's the question, how it really responds to.
yeah.
But that's where that no one can answer. Not yet.
I really enjoyed your talk. It's great. I was wondering, you showed this end Cretaceous
Yeah.
event,
Yeah.
and I was wondering how that impacted on the various groups that you've studied in the present day. Can you, from the genomic data, see how, for example, the whole Archaic kingdom, how they responded at the end Cretaceous? Was the genomic data not
So that's
specific enough for that?
actually this is some really hot topic what people are working on these days, and I'm doing that as well. So what you would do now, you would look for different situations that are similar to the Cretaceous situation in the modern ocean, do a metagenome, and then you would need to deduct biomarkers from that, like lipids or pigments that are more stable and go into the sediment records, and which you could then recover. And via this kind of indirect approach, which I personally think is very valuable, you could try to track it back. And that's what people now try, that the metagenomic data sets are getting better and the evaluation pipelines are more specific. But it's not done yet. So there's still some room to do.
Plenty to
things.
do, yeah?
Yeah, there's plenty to do, sure. But yeah, that's one of the hot topics actually.
There's some more questions? Let's give Carl a